Hi, everyone. Welcome to this live broadcast. So I'm Karim Beguir. I'm a member of Deep Learning Indaba's steering committee, and very excited to be moderating this event today. So we have an exciting lineup of speakers. But first, I wanted maybe to introduce today's topic, tell you more about the Grand Challenge and how we arrived to the point at which we are now. So I'm going to share my screen and we can take it from there. So you should be seeing my presentation now. So as I said, super excited to welcome you to the Indaba Grand Challenge. Maybe I'll say a few words as we have many people from across the world that are joining us for this live stream. I tell you a little bit about Indaba. For those who don't know it, Indaba is a Zulu word that means gathering. And deep learning Indaba goal has been to strengthen machine learning and AI in Africa. And we do this through multiple initiatives, coming from like teaching, research, networking, sort of like being a catalyst for the machine learning ecosystem in Africa. And we do this through multiple initiatives. First, we have the annual Deep Learning Indaba, which rotates between the different countries in Africa. We also have national events in many African countries, which we call the Indaba X's. And finally, once a year, we distribute prizes, the Kambuli and Mathai Awards, which are to reward and distinguish like unique contributions to the African machine learning ecosystem. Incredibly, Indaba has been getting significant traction. So when we look at where we were in 2018, we had 13 different national events across the continent, 1,000 plus attendees. We accelerated in 2019, covering a total of 27 national events across the continent, 2,800 plus attendees, with the main event happening in Nairobi in Kenya, which was a large attendance as well, around 500 to 600 people. This year, as you know, we are delaying the national event. It will take place in Tunisia, probably in summer 2021. But importantly, this is not a reason not to progress machine learning and take initiatives. And this is in this spirit that we've built the Indaba Grand Challenge that we are talking about today. So what is it about? Here, the main focus is drug repurposing. If you look at, the way drug development works, it's kind of a shame, but it's really focused on first world diseases. So literally, large pharma companies are going to be putting a priority where it makes sense from a financial perspective. But that means that you have many neglected tropical diseases, what we call NTDs, that affect a crazy amount of people. We're talking about 1.5 billion people across the globe being affected, 500 million children in 150 countries. And it's not like necessarily that these diseases are impossible to solve. In many cases, actually, there might be a solution might exist somewhere, but we don't know. So if you look at all the different medical compounds that we know are safe, that have been developed for other diseases, there is an opportunity that they could work for neglected tropical diseases, and yet we don't know. And this is exactly our goal today. It is to focus on one disease, Leishmaniasis, which is a parasitic disease that kills 50,000 people a year, which is enormous, and kills from people from South America to Africa to India and beyond. And we actually believe that combining bioinformatics with machine learning and with available public data, well, there is a hope that we can crack those type of diseases, develop potentially good candidate cures that will then go to in vitro testing and ultimately clinical tests. So we believe, there is no better use of machine learning than to save lives. And, this is an effort which we think, is quite important, and that, the Indaba community is very well positioned to tackle. So if you look at, at the grand picture, and we will be, having several speakers, getting us into the detail of the challenge, but really, what is our goal here? Our goal is to literally save lives. There is nothing more important that we could do. There is no better use of machine learning than to save lives and empower our community, in the same way. So, the real idea here is always the same. AI is a very powerful technology, but it must truly benefit everyone. And that's not necessarily the case by default. In the world in which we live, we see that, fairness is a significant issue and it is in no way guaranteed. And by doing and taking initiatives such as this one, I believe we are being forward-looking, we're building a better world, and importantly, we're saving lives. There is also something quite interesting about this challenge, is there's a tremendous amount of data which is already there. So, the idea is there's so much data that is probably beyond, a small team to cover. And so it's actually a great problem to be looked at from a distributed and community point of view. And at Deep Learning Indaba, our motto is Masakane, is we build together. So by collaboration, we can make, we can tackle like really big challenges with fresh ideas. And we actually believe that our approach in this particular case is innovative. Most impactful projects in the world tend to be driven by governments or large corporations. Here, our approach is pretty much bottoms up and unique on its kind on the continent and beyond. We focus on the community. We leverage the help that we can get from African startups and other tech partners. And we believe that this is a fresh approach that can be very fruitful into this problem. So I really want to thank our partners that are helping make this possible. And as I mentioned, we have partners from across the world, but we also have African startups such as Zindi or InstaDeep, like literally building up the tools and platform that make this possible. We want to thank also IDRC from Canada, SIDA, and Knowledge for All, that are helping provide funding for this competition, which is quite important. So there will be an opportunity to get funding and applying for grants. And we will have, Kathleen later on speak about this. And finally, we also want to thank our support partners, such as the TensorFlow research team, which is going to be providing like cloud compute power with cloud TPUs, but also our community partners, such as Data Science Nigeria, Data Science Africa, Black in AI, DSNA, Alliance for AI. We believe that we are stronger if we work together towards the same goal. And this is an ambitious goal, but we believe it can be achieved. So let me introduce maybe our speakers. So we have an exciting lineup today, starting with Benjamin Perry, which is from the DNDi organization, and he will be speaking. Like DNDi means Drugs for Neglected Diseases Initiative. So this is at the heart of what we are trying to achieve today. Elaine from Boston University and Data Science Africa will be explaining why machine learning is so important for data science and public health, and what are the opportunities to deploy machine learning in novel ways in this area. Marcin Skwark from InstaDeep is a bioinformatics expert, and he will be explaining to us how the challenge works and why this crazy moonshot is actually possible today in 2020. Celina Lee, who is the CEO of Zindi, will explain how participants will be leveraging the Zindi platform to make this possible. And finally, Kathleen will be discussing how to get funding. If you are serious about this project, there is an opportunity to get funding, and up to $8,000 per team, depending on the applications we receive. And she will be detailing this a bit later today. And finally, we'll organize a sort of panel to ask questions to our speakers and get you as much highlights as possible. So thanks a lot for joining us today. And we can get started with Benjamin from DNDi, who's going to tell us more about tropical neglected diseases. Thank you.
Thank you so much, Karim. I hope you can hear me.
We do.
Hi, I'm Ben Perry from the Drugs for Neglected Diseases Initiative, as Karim just mentioned. I'm actually based here in Switzerland, but DNDi are very much a global organization. We have quite a large presence, particularly in Africa, where we have offices in Nairobi and Kinshasa and also in Cape Town. And we're involved in research and development in neglected tropical diseases, and Leishmaniasis in particular is a disease that we're heavily focused on. So I'm going to give you a little bit of information about the disease itself and a little bit of information about what the landscape looks like in terms of available drugs for this disease and where we want to take that landscape and what we want to eventually achieve as a community working on this disease. I myself am a drug discovery scientist. I'm a chemist by training. I know a little bit about this disease. I've been working in it for about 4 or 5 years. And I also have an interest in AI and deep learning at a layperson's level and implementing that in drug discovery. So I'm really excited to be part of this discussion. So a little bit about Leishmaniasis. Leishmaniasis is actually a family of diseases caused by the Leishmania parasites. There are about 20 different species that are responsible for these diseases. The first one, and probably the one at the forefront of our mind when we're talking about research and development, is visceral Leishmaniasis. I'll talk a little bit more about that in a second. But there are also other very prevalent forms, particularly cutaneous Leishmaniasis and mucocutaneous Leishmaniasis, which are more skin disease issues, and also post-Kala-Azar dermal Leishmaniasis, which is a hot emerging topic in terms of the Leishmaniasis field, because it seems to be a reservoir for various Leishmaniasis parasites during the occasional troughs and peaks in the wave of the way that these diseases progress. I'm going to talk a little bit mainly about visceral Leishmaniasis in this background, although all 4 of these areas of Leishmaniasis, I think, are of interest and in dire need of improved treatments. So visceral Leishmaniasis, it's the most severe form. It's caused by 2 particular parasites within this genus, Leishmania donovani and Leishmania infantum. And there are hundreds of millions of people at risk worldwide, about 82 countries that visceral Leishmaniasis is present. It's transmitted by a sandfly vector, so very similar to the way malaria is transmitted by mosquitoes. Leishmaniasis is transmitted by sandflies. And the symptoms for visceral Leishmaniasis are pretty severe. We're talking about a prolonged fever and an enlarged spleen and liver, anemia, weight loss, and most importantly, this is a disease that is fatal if there is no treatment. There are tens of thousands of new cases of VL every year. About half of those are found in East Africa. I think Karim mentioned that this is a disease that's prevalent across the globe, particularly Brazil and India outside of Africa. But East Africa, South Sudan, Kenya, Ethiopia, this is a real problem. There's also a major problem of co-infection with HIV, again, particularly prevalent in East Africa, which exacerbates both diseases and is really something that needs to be tackled quite quickly. And there's a pediatric element to this, so there are about 50% of the cases for this disease are in children. Now, there are treatments available for this. I'm a chemist, so I love this slide. I appreciate that not everyone is a chemist and might not get those structures up there, but the main important point on here is that there are drugs available, and as you'll see across the top, we have the good, the bad, and the ugly, and the only place you see green on this slide is where it says the good, nowhere else, because these drugs are pretty bad. They are not very nice to administer, they are not very efficacious, they are toxic, they have terrible side effects, and in some cases they're have issues with affordability. So most of these 4 of these 5 drugs require slow IV or IM infusion. They require hospitalization for many, many days, in some cases weeks, painful administrations, and side effects that are barely tolerable. One of these products, the one on the right, miltefosine, is orally available, so it can be given as a pill, but it's teratogenic, so it can't be used in women of childbearing age. It also has some pretty awful toxicological side effects associated with it. There is also a big problem with the efficacy of these drugs. So this is quite a detailed table. I'll hopefully be sharing my slides later. But what this should show is that depending on the region of the world where you investigate these treatments, some of them work very well, particularly in Asia and in India. You'll see some of these treatments work quite well, but others, and particularly in Africa, there is a really poor response rate. Even some cases as low as 50% success rate, which is not good enough. So there is a clear need for a new treatment. We need something that is safe. We need something that is efficacious. We need something that is cost effective and cheap, and ideally orally administered. So a huge need for new drugs for leishmaniasis. We've mentioned it's a global disease. You can see here in the dark brown the main countries where VL is prevalent, but also it is found in other countries. And another key point from here is that, as on the previous table, the treatments that are used in different areas are different because different treatments work in different areas. And in East Africa, the first-line treatment, which is SSG and PM, which is a 10% failure rate, again, not great, is not the same as the treatments that are used in Brazil or the treatments that are used in India. It's a very complicated disease. And as I pointed out earlier, I think 90% of those 50 to 90,000 new cases are found in just 7 countries, Brazil and India being 2 of them, but the other 5 being found in Africa. So at DNDi, we've been working on this disease pretty much non-stop since our founding in 2003. Our strategy is to develop new treatments, and we're very interested in this and interested in working with partners who are interested in this. 15 years ago, the situation was really quite dire. The treatments that were available were very, very toxic and painful. There were some that weren't registered, so not controlled. It wasn't known what you could and what couldn't use, and there was an expense problem. We've started to introduce new treatments slowly, 2010, 2011. We have some that are available in certain countries, but again, these are still not great. What we really need are new drugs, new treatments that can be given orally and that are safe. And we've been working very hard on that for a while. So our overall goal is primarily on visceral leishmaniasis, but we are focused also on cutaneous leishmaniasis. And I think that the approach that DNDi takes is very similar to that taken by other players in the field. And there are other organizations like DNDi who work in leishmaniasis. In particular, there are 1 or 2 big pharma who do work in this space. So Novartis and GlaxoSmithKline particularly are active in this space because through their global health initiatives, but they work in collaboration with DNDi. So when they actually arrive to looking at clinical work and treating patients, they do that in collaboration with us. And so we're all focused on the same sort of thing. And what we've agreed is that the current treatments, mostly injectable, toxic, not particularly efficacious, limited spectrum, meaning that they work in some countries and not in others, in some patient populations and not in others, and they're expensive. And what we want is to turn these into compounds, into treatments that are oral, well tolerated, no side effects, short duration, 5 days, maybe 1 tablet a day, efficacious everywhere, and affordable, and can be registered in the health systems where they're needed to be registered. So in Africa and Latin America and in parts of Asia. So we've been working on this for quite some while, and this is a snapshot of where I would say the global landscape currently sits. There are a couple of potential treatments on the way from outside of the DNDi organization, but this represents a substantial chunk. It includes compounds from GlaxoSmithKline and others. And this is a drug discovery pipeline. You'll see this in any pharmaceutical company's website. It shows the things that they've been working on in early discovery, the early projects on the left, and and on the far right, implementation of new treatments. And the clinical phases, phase 1, phase 2, phase 3, where we're looking at safety and then efficacy in patients, and then detailed efficacy in patients in phase 3, and ultimately registration. And this actually looks quite healthy at first glance, particularly to maybe a naive eye who might look at this and say, wow, there are a lot of potential new treatments on their way. Each one of these boxes is a new treatment. But the reality is that this is nowhere near as rich as it needs to be. So just this space here, the preclinical phase 1, so this is when we have maybe found a molecule, a new chemical that we think this could work in humans. Let's start to see how it pans out if we give it to healthy volunteers. Is it toxic? Does it get into the bloodstream? Does it get where it needs to go? We currently have about 7 or 8 products that are in that space. The thing is, we need about 20 there. We're not even halfway. In order to get 1 molecule to patient, we're pretty certain, based on known attrition rates in this field, we need 20 shots on goal here to get 1 successful goal. So we need to keep filling this pipeline. We need to keep putting new molecules in here, and hopefully that is somewhere that this grand challenge can have some impact. So I was having a little bit of a thought about how we might be able to link up AI, deep learning, with Leishmaniasis research and discovery. And I've got a couple of ideas. These are just off the top of my head. The obvious one is in discovery of NCEs, and I think we'll hear a little bit about that later. NCE is a new chemical entity. It's a new chemical that could have an impact on the disease. We could think about identifying new biological targets and ways that the parasite behaves, or proteins that exist in the parasite that we could tackle, try to knock out, or try to enhance in order to then kill the parasite or prevent it from replicating. And we can also think about using AI to maybe identify new starting points against those targets. And then there are other approaches, the repurposing, again mentioned by Karim, this idea that maybe we can take existing drugs for other diseases and think, well, could this possibly have an impact? We know how it behaves in the system. We know how it behaves in humans. We know how it might behave against other parasites or other infectious pathogens. Could this work against Leishmaniasis? It's a huge amount of data. It's very, very complex to plow through that data. So there's a clear area that a deep learning approach could help out. And also the idea of combination therapies, not just one new treatment, but what happens if we combine a new treatment with an existing treatment or two existing treatments. Again, the amount of data here and the amount of possibilities is huge. There are also opportunities, I think, in looking at clinical and diagnostics, thinking about how you can maybe diagnose patients, particularly in cutaneous leishmaniasis, which is a dermal disease, and so we have images of the lesions, and you can imagine, maybe ways that imaging and image analysis could be used to maybe make some breakthroughs. And then there's also, at the other end, the epidemiology and the idea that, how do migration patterns amongst populations at risk impact the epidemiology of the disease and the prevalence of the disease, and the way that resistant strains might move from one place to another or might rise and fall, and how is that going to be impacted, for example, by climate change, and can we predict maybe how resistance is going to emerge to new drugs? So right at the other end of the spectrum. And then the last one here on the right, this is this idea of understanding the biology behind the disease. One thing we know is that you can have a chemical that works really, really well at knocking out a particular protein that is critical to the Leishmania parasite. So you know that if we knock out this protein with this chemical, the parasite should die. But when you actually test it against the parasite, nothing happens, because it's not just that the molecule, the chemical, needs to tackle that particular piece of machinery. It needs to actually get there. It needs to get into the cell that the parasite is hiding out in. And we don't fully understand how that happens and what is needed for a particular chemical to get to its target, to get to the target parasite. Again, a huge amount of data available and a really interesting area for intervention. And a couple of other ideas. I've put this, another imaging option down here. We use imaging in biology all the time, looking at how we can kill parasites. We have a huge database coming from pharma company libraries that we've screened, and coming from our own efforts. Millions of images of parasites treated by compounds that can kill parasite, and seeing how those are affected and how the cell morphs around the parasite, how the parasite disappears. Is there something we're missing by examining those images by eye? Again, I think another classic AI opportunity. So I think that was everything that I wanted to say about the background to AI, to Leishmaniasis, and how I think that AI could work. So I'm now going to, I think, hand over to the next speaker, who I believe is Elaine.
Yeah, thank you, Ben. It's amazing to hear from you, and to see that there is so much to do still on those problems. So the interesting point now is how we combine this with machine learning and AI. And so Elaine, who's from Boston University and Data Science Africa, will give us sort of an overview of what we could do with machine learning on those type of problems. Elaine.
Sorry. Thanks, Karim. So Benjamin already talked about some of the applications of machine learning to the specific problem, and I'm going to give you more of an overview of a broader view of how machine learning can be used in public health and epidemiology. So I like to start off with definitions. So public health is the science and art of preventing disease, prolonging life, and promoting health through the organized efforts and informed choices of society, organizations, public and private communities, and individuals. So this basically indicates that public health is basically everything, whether it's housing, transportation, how communities are built, what we have access to, the policies that are being made by the governments, and how all of those things affect our health. And when we talk about epidemiology, we're usually referring to the study of the patterns of disease, injury, and health, and factors that contribute to these patterns in specific populations. So specific populations is important, including that is very important because the patterns that you see in one population are always the same patterns that you see in another population. So neglected tropical diseases, for example, are mostly found in the global south. And so when you talk about those diseases in the West, people might have different understanding because they've not seen those diseases or are not exposed to them. Risk factors can include everything from the social factors, so how different people affect us and impact our health, to environmental factors, so things like air pollution, and also what we have access to in the built environment. So do we have access to good schools or housing, or do we have access to places where we can exercise, we feel safe in our communities? So all of those things can affect our health. A lot of what I do is called digital epidemiology, so I tend to focus on how we can use digital technology and data from places like social media and other internet platforms, so from search data, business review platforms, customer perspective, and different things, and how that can inform us about health in different communities, globally. So some of the opportunities for machine learning applications in public health and epidemiology include surveillance of disease, risk factors, and medications. So these can go all the way from looking at developing tools that can be used in hospitals for monitoring diseases, to having access to digital data streams and trying to understand how those can inform health, to developing platforms for crowdsourcing, so having people participate and provide information about what's happening in their communities. And this can be developed as mobile apps or websites or other tools. Risk factor surveillance, as I mentioned, include both social factors, behavioral factors, like whether you smoke or drink. These are all things that affect our health. And I think there's a lot of opportunity for medication surveillance, especially in Africa, where fake medications contribute to a significant number of deaths from different diseases. And there are people working in that space. They've developed AI tools that are supposed to detect unsafe medications to prevent the number of people that actually die because they buy a medication thinking that is actually the right medication, but it's actually fake. So machine learning applications can complement traditional approaches to disease monitoring. So what I mean here is that when we develop machine learning applications, it's not always because we want to substitute what is already being done. In a lot of cases, we can use it as a way to complement what already exists. So for example, during the current coronavirus pandemic, there's a lot of traditional approaches to public health that are being carried out. These methods have been used for a long time, and we know the work. So machine learning approaches can then complement those existing approaches. For example, contact tracing. How can we use machine learning in an ethical way that can complement existing contact tracing approaches that public health practitioners have been using for a long time? Machine learning can also be used for public health communication. So this includes understanding public sentiment towards different public health issues and interventions, so things like vaccines. So what do people think about vaccines? I think this is very important, especially right now as we think and talk about the coronavirus vaccine. We know there's already a lot of negative sentiment towards that. So understanding what people think and how they feel towards something like vaccines can inform public health practitioners on how best to educate the public about those particular interventions. We can also think about monitoring misinformation and also information seeking patterns. So what are people looking for online, and how can these inform us about people's needs? So on the right, I have a visualization that was developed by Maureen and Mutorin, who's also part of Data Science Africa. And we wrote an article that looked at questions that people were asking in 21 African countries to see how have questions changed over time and how can these questions be used to better inform public health interventions in those countries. We can also use machine learning for disease diagnosis, prediction, and clinical management. So there's been a lot of work around diagnosis, especially. So AI can facilitate rapid disease diagnosis for different diseases. And as Benjamin mentioned, there is an opportunity here to look at Leishmaniasis detection and diagnosis. You can also predict disease progression within individuals. So how do individuals go from having mild symptoms of a disease to having more severe symptoms over time? You can predict temporal and spatial spread of diseases. So looking at cases that have been reported and then predicting over time how that's going to change at the temporal scale, but also how that would change at a spatial scale. So if you look at a particular country, how do the rates of disease differ from one region to another, and what are some of the factors that are predictive of those differences? And of course, there's drug discovery and development. So on the right is an example of a study that I did many, many years ago. And this was to look at how different diseases affect genes similarly. So are there diseases that have similar impacts on our genes? And could that inform us about the kinds of medications that we use for these different diseases? Can we repurpose some of the drugs that we're already using for a particular disease for another disease based on what we know about their effects in the body? Of course, there are always challenges. So when we think about machine learning, we think a lot about data. So what is the data that we're using, and how would that data affect the results that we produce? So we have biased data, we expect to produce biased results. And then there are also issues around privacy. So when you're using data, whether it's from a clinical setting or from the internet, what are some of the privacy concerns that you should be thinking of, and how can you make sure that you're not exposing people to more risk than they already have been exposed to? Thinking about data representation. So just because your data set is big doesn't mean that everyone is represented. So who gets left behind, and why do they get left out of that data set? And what can you do to supplement the data, especially when there are individuals that should be included that are not being included? Incomplete data is also a challenge, especially when we think about certain diseases in developing countries. Data collection might not be the way it should be, and so we have to think around that and think of how do we develop better data standards. How do we also supplement missing data when that data is incomplete? Systemic issues linked to data collection, analysis, and reporting are things that we should always be considering. There are certain populations that get left out of data collection efforts. And because they're left out of the data collection efforts, when we do analysis and make decisions, the decisions we make will not impact them positively. And so it's always important to think about who collected the data set, how was it collected, what are some of the limitations in that data before we actually use the data for decision making. And lastly, I think it's always important to evaluate the systems that we're building in clinical settings, especially when we talk about systems that are supposed to affect the management of diseases in hospitals. So things around disease diagnosis. So it's not okay to just develop an AI system that you think works without actually going into a hospital or into a clinical setting and testing that system to make sure that it works the way it's supposed to work. So for example, it might be able to diagnose disease better, but what are some of the other problems that it creates in that settings? And is it okay for those problems to exist, or is there something that you can do to change your system so that it's not just addressing one problem, but can address multiple problems at the same time? So I think that's all I have. Thank you guys for your time.
Thank you, Elaine, for the comprehensive overview of uses of machine learning in the healthcare area. I think we talk a lot about indeed bias in machine learning and AI, but in this particular case, it's a matter of life and death. You have also issues with data privacy, and at the same time, we need to progress and use machine learning to solve those problems and save lives. So I think that's very interesting debate, and I'm sure we'll have questions when we get to the panel side of this live stream. So after this overview of machine learning and AI, we're now going to do a deep dive into the grand challenge itself, the science behind it. And so Marcin, who's been instrumental in building the tools that make this grand challenge possible, can give us like an in-depth look at it. Marcin.
Hello, so I'm Marcin Skwark. I work at InstaDeep. I'm a computer scientist turned life scientist, I guess. And I would like to tell you about some of the exciting things we're going to do together. First, let's get the formal thing out of the way. How can you win the challenge? We can speak about winning. So what we want you to do is to submit the structures of the protein and the drug that we believe that binds that protein, and then we will take it from there. So if you submit a structure with the drug or the docked place where it's supposed to be about, we will evaluate it. But otherwise, you don't have to worry about it. As long as the molecule that you believe is the appropriate drug and the protein that you believe the molecule targets are in the file, you're good to go. We will provide the scripts that will generate these input or submission files for you if you have, for example, just the protein ID and the drug ID. However, we have some evaluation criteria. First, well, I regret to tell you, the submission needs to be relevant. We accept only submissions which actually target Leishmaniasis proteins. The second, the submission needs to be novel. So we have an ongoing process that checks the literature, the positions for new anti-Leishmanial drugs. So if you submit something that's already published, that won't really work. And finally, the drug that you submit should bind to the target, and that's something that we'll assess by the computational means. And ideally, we would like the drugs to come from the small set of already approved compounds, compounds that are known to be safe and efficacious for some of the diseases. There's around 6,000 of such drugs. They are all present at Zindi's website. But if that's not possible, we would prefer the drugs to be purchasable, the one off the shelf. There's a set of first we would like them to be tested, then we would like them to be purchasable. There's 114,000 drugs that have been tested, and there's 12 million drugs that can be purchased. So there's quite a bit to select from. But in order to submit something, you actually need to have something to submit. And let me give you a very, very short crash course on drug discovery. Pretty much all that happens in the living organisms is due to proteins. Proteins bind their partners. They take some small molecules and they use them to perform some functions. And as such, and they do it by spatial 3D interactions. So if you provide a protein with an attractive partner to bind to, it might bind it instead of its natural partner. Or if you provide the protein with something to bind to the interaction surface, it will not bind the other protein that it's supposed to interact with, because there will be something hindering it. So the chemicals that bind with high affinity to proteins are really feasible drug targets. The other thing is that, well, we know a bit about Leishmaniasis, and we know a lot about everything else. And good news is that nature reuses components. And so if your organism of choice has a certain protein, you could be almost sure that the very similar protein in a different organism behaves the same way, responds to the same drugs. So there's plenty of opportunity to learn from the data that's already there. And The other thing is that there's nothing special about drug binding. It's really a lock and key mechanism. If your protein has residues, has positions that bind your drug, bind the natural component well, if you design something that binds better, it will bind. There's nothing secret about that. And we have plenty of data, as I said. There's in PubChem, one of the big databases, there's over 103 million compounds, nearly 300 million data points on what binds where. So we can definitely do something. One of the things that we can do is we can look at the similarity. The first thing is, well, I told you that the similar proteins act in a similar way, and as such, you can either compare the proteins by the amino acid sequence, or better still, by their structure. We're trying to make it as easy for you as possible. So we're providing the models and experimentally determined structures of proteins ongoing. So there will be a Google storage repository in which for most of the Leishmaniasis proteins you can get either an experimental structure or our best guess at that structure. If you have that, you can then start computing different metrics. You can look at the binding pocket of such a protein and try to find other proteins that have similar binding pockets. You could look at the ligand, the small molecule that's bound there, and if you know that it binds to this target, you can try to find other ones that look kind of similar to it or have similar features. The features that make for the small molecule binding are not obvious, but it's once again something that can be learned from the data. And finally, what Ben has suggested is that I'll move closer to the microphone. What Ben has suggested is that that we would like to find new drug targets, and therefore, if there is a protein that already is a target of a drug in a different organism, and there is a similar one in your organism, perhaps you could try to use that as a starting point. The other thing that you could look at is the interaction networks. The living organisms share not only the machinery, the proteins, but also the whole operation plan. So the same processes in most of the organisms are performed by the very similar combination of proteins. There are so-called pathways and interaction networks, metabolic networks, and all of these have been painstakingly figured out. They are all available in a machine parsable way, and you can just take them and try to go from there. There's plenty of opportunities here. So even though I tell you there's plenty of opportunities, I know it's kind of difficult to know where to start from. So the first thing is that then we provide you with the protein sequences and protein structures, and you could try to use standard bioinformatics tools that we will be linking to on the website to find similar proteins and perhaps cross-relate them with the known activity assays, so the experiment records. And then if you could also try to find the known targets in an organism, then you could perhaps start from the other end. We know some of the anti-leishmanial drugs, and you could try to find out what are their targets in the organism. We know very little about that, but it's something that once again can be learned from the data, or at least we can estimate it to a certain degree. So you could also try to find similar drugs. All of these can be done with standard cheminformatics methods that we'll be trying to teach you over the course of the next few weeks. But also, you could try to think about different data representations, learning some sort of embeddings, perhaps, that would allow you to find distant relatives that still do the same thing. And furthermore, as Karim, as Ben, as everyone mentioned, there's loads of data, and I mean loads and loads of data. And on top of the data that's in the databases, there's data in free form. There's data in papers that you could then employ your NLP prowess on. And hopefully that could give you an insight as well. So we have here we have a small sort of user journey of what could really happen. Amara, let's assume that Amara wants to learn more about Leishmaniasis and perhaps find a new drug. First, she goes through the literature. She finds the sort of some grasping points on what she could start. Then she goes through the public data repositories and realizes that there's this whole class of drugs that seem to target the same mechanism or the same protein. And she built a small, or not that small, machine learning model that helps her find more drugs of that type. And then she tests them locally, submits them to Indaba, where they get tested on the equal footing of every other submission. And, well, having this score that she gets, well, repeats the whole process. In such a way, she may be able to find drugs that bind better than anything else that has been found before. And the beautiful thing that separates this endeavor from many other similar to it is that the drugs that we find, the ones that are prospective, will be experimentally tested at DNDi. And therefore, any submission that you make can be actually really life-changing. It can be something that today is just bits and bytes. Tomorrow, it could actually be in a doctor's purse somewhere in Africa. And well, I hope that you realize that it is a moonshot project. We're trying to tackle a disease that's caused by 15 different parasites, or actually 20 different parasites. One that's heavily underfunded, heavily underdocumented, but what we have the data behind us. We have lots of statistical machine learning models that we can apply as well, and we have enthusiasm of the community. If you look at the recent advances in computational life sciences, be it bioinformatics or cheminformatics, very little of them came from biologists or chemists. Most of it came from computer scientists, from statisticians, from people who were enamored with the amount of data that's there, and decided to do something exciting with it. And I believe that together we can really make a difference. We can put the pieces together in a way no one has done it before, and hopefully come up with the best solution to a global scourge that Leishmaniasis is. Okay. Thank you so much for your attention, and I hope that I got you at least a bit excited.
Yes. Actually, Marcin, a question from the audience is, most of our audience is, more into machine learning than bioinformatics, and so is mostly beginners. So, will you be providing sort of like initial courses or things that can help accelerate understanding for this challenge?
Yes, you're not the only ones that would like to make a difference, but seem to lack the toolkit. We will be providing the links to very good courses and tutorials on how to do all of the things that you need to do. And I believe if there's sufficient interest, we will organize some sort of live tutorial sessions in which I'll go through most of the tasks that you may want to do for this particular problem. And we will also provide, hopefully, as much of support as possible to anyone who gets stuck.
No, that's a great idea. I think the live tutorial will be an awesome opportunity to spread the knowledge. And you know, if you are new to this area and this sounds complicated, it's absolutely normal. But you will see that, you know, once you have a few of the building blocks, it becomes, you know, much easier. And on top of it, you know, we've spent time to make sure things are easy to perform from a platform point of view. And so I'm very excited to welcome Celina, the CEO of Zindi, who is going to explain to us how the competition works in simple terms on the Zindi platform.
Great, thank you so much. I'm going to go ahead and share my screen. Yes, my name is Celina and I'm the CEO of Zindi. For those of you who maybe haven't heard of Zindi yet, Zindi is a data science competition platform focused uniquely on the African market. We host a community of over 15,000 data scientists dedicated to solving some of the world's most pressing challenges, such as this one, through machine learning and AI. And we really created Zindi with the realization that organizations across Africa and be them companies, governments, are generating more and more data. And at the same time, there's this growing pool of talent in Africa. And I think that's why we're so thrilled to partner with the deep learning Indaba for this project, because I think we share the vision, we share the excitement around the growing pool of talent that's coming out of the continent. Data scientists can join Zindi, and with that, they're able to really start to hone their skills on new problems, such as this one, build their skills on real-life data sets for real companies and organizations that need the solutions. They're also able to start to connect with each other, meet mentors, and even companies and organizations that are looking for talent like theirs. And at the end of the day, those Zindi's, so I'm dialing in from South Africa. Zindi is a African startup company, but we're also a social enterprise, because at the heart of what we're doing is our mission to build the data science ecosystem in Africa. And we're really driven by the belief that AI should be for everyone, that AI should serve everyone, and it should also be a viable, accessible career path for anyone who wants to pursue it. So I mentioned on Zindi that we tackle some of the most pressing problems. Anything from, if you join Zindi, you can see competitions for anything from child protection to sea turtle conservation to roadside accidents in Cape Town. And so that's why we're really excited to host this challenge for the deep learning Indaba. So I'm going to take you through a bit of just the very practical steps on how you can join this challenge, because I'm sure after hearing everyone else speak, you're ready to get your hands dirty. So the first thing you have to do to join the challenge is to visit the Deep Learning Indaba site. And with this, you can register, right? You can register for the challenge. You have to register for the challenge in order to join the challenge on Zindi. So this is really your first port of call and your first step. If you already have a Zindi account, you would enter your Zindi handle. And if you don't have a Zindi account, you can actually create a Zindi account through this site. So it's all one step. Once you've registered, you'll receive a password to join the challenge. And so you can actually then go to the Zindi site, and I'll just show you. If you go to the competitions on Zindi, hopefully, let me know if you're not able to see my screen. I'm hoping you're able to see my screen on here. If you go to the competition site, you can find just search for the Indaba Grand Challenge. And here you'll join the competition, and you'll be prompted to enter the code that you got from the Deep Learning Indaba. Once you've joined, so Marcin went through some of the data that you might be using. I'll also say that right here on the site, you have the user journey that he described, so you can get some idea of the steps and how you might even start the challenge. And we also here have some of the data sets that he might have mentioned, and where you can start, so you can actually download the data sets and start building your models. Once you're ready to make a submission, you'll actually be able to make a submission here on the site where you'll upload a PDB file. And so all of these things are explained and described here. And what's nice is that if something is still not clear, if you're still trying to figure out what are what are the resources that you should be looking at? What are the first steps? We also have a discussion board that can get quite lively. So as you join the competition and as others join the competition, hopefully there'll be a lot of dynamic, ideas shared here and, help and questions and answers going on the discussion forum. And the last bit on this competition, which is always fun, is as you make submissions, you'll actually be given a score. So there's actually a scoring algorithm that's sitting behind this. And you're going to be trying to maximize the score that you get, and you'll be placed on this live leaderboard based on the score that you achieve. And you'll actually be able to make up to 20 submissions every day. So you can continue to work on improving and refining your models. Let me go back to my slides. Sorry. I'm confused which, well, let's just, I'm sorry about that. Now I lost which window I'm in. But I just want to close by saying that I saw a lot of familiar names on the webinar today, and so welcome. It's nice to see some Zindians here. And for anybody who's new to Zindi and hasn't joined before, welcome to the community. I think that it's an exciting step, and it's a very dynamic community. And I think that together, as Marcin said, together we can do big things. So I'm looking forward to seeing you all on the leaderboard. Thank you very much.
Thanks a lot, Celina. And I think it's super exciting that we now have basically a platform where, something that sounds like so complicated as such as like curing a real life disease, and a lethal one, can actually be potentially solved just by making submissions, no matter where you are, to the Zindi platform, and working with the API and the platforms we've been developing with all the volunteers. So this is in many ways groundbreaking, and we're super excited to see what's going to come up from this. I want to also mention two things: is that this is actually a challenge which we mean to be very open and very collaborative. So please make sure you use, the discussion boards. But in particular, there is no private leaderboard. This is not a competition where the goal is just to be number one. The goal is actually to crack the problem and save lives. So everything is going to be public. You have a public leaderboard. We encourage, collaboration between the teams. And the goal is really, as noble as it can be. But at the same time, of course, we also have opportunities for the teams that want to pursue this challenge, in the most serious and focused manner. There will be funding opportunities, and we're very excited by bringing this opportunity to our community. And so Kathleen, who is basically from AI4D and also Deep Learning Indaba, is going to tell us more about how you can get funding for this challenge.
Thank you, Karim. So, like Karim has mentioned, I will be speaking briefly about funding your ML research, particularly if you would like to participate within the grant challenge, using that as a research project, or also more generally if you have ongoing projects or are interested in working on something different that can potentially be funded. So my objectives with the presentation are to tell you a little bit about the funding opportunities we have available, go through eligibility, applying for the funding, and also some mentorship avenues that we have set up. So this is a call for proposals for funding for mini projects. And when I say mini projects, this could be new work, or it could be existing work. It may be a small project, or it may be a segment of a much larger project. Really, our categorization when we say mini project pegs this on the fact that we would like the work to be completed by January of 2020. So like I have mentioned, this would be a great avenue to get the research that you're doing via contributing to the grant challenge funded. But also if you are working on work that could benefit from funding or have other ideas that you would like to implement, feel free to prepare a proposal and send those in. Again, the caveat is that it is work that can be completed by January 2021, next year, not this year. The proposal submission deadline is 6th of July, which is pretty soon, and awards will range in value from 2,000 to 8,000 USD. And I'd just like to quickly take this opportunity to acknowledge the sponsors that have made this possible. That's the International Development Research Center, the Swedish International Development Cooperation Agency, the Knowledge for All Foundation, and all this is made possible through the AI4D initiative. So work that is eligible. Ideally, it should have the Sustainable Development Goals in mind. So the SDGs are developed by the United Nations and are a call to action for all of us to promote prosperity while protecting the planet. There's 17 of them, which you should definitely check out. I would suggest thinking through which goal or goals your work potentially touches upon. And the Indaba Grand Challenge is a great example of work tackling goal number 3, which is good health and well-being. And we hope to see more examples like that of AI that is used for development. A full list and a more detailed write-up of eligibility criteria is listed on our website. But then other things that I think it would be important to highlight here is that this needs to be work that is conducted in Africa. I think it goes without saying that it should have a strong machine learning, artificial intelligence, or data science component involved, and that it should reach the deliverable by January 2021. So yeah, I got the date right on this slide. In applying for funding, we recognize that the African AI ecosystem is very diverse. So not everyone who will be doing this research is situated at an academic institution. So we have prepared several options for funding. The first and preferred avenue is, of course, getting your funding through an academic institution. If you're affiliated to one and can receive the funding through that, please apply via your academic institution. Alternatively, you can also apply through an affiliation to a charitable organization. So that's if you have someone on your team that has worked with, has an affiliation to, or is perhaps, or if the charity is perhaps a partner on the implementation of your work. In addition to those two avenues, we do understand that in some instances, these options will not be ideal. So you potentially are affiliated to a university, but then you think that process may take too long in terms of actually receiving your funds. So if it's a situation where delay in receipt of funds would actually negatively affect your project. For example, if you have contractors who need to be paid in a timely manner to deliver some aspect of the project, you can opt to apply as an individual. So you can receive the funding through a project lead. In this instance, the project lead would enter into a contractual agreement with our organization, and they would have the funding sent directly to them. So the organization in this case is the Knowledge for All Foundation. And if you opt for this case, you will have to justify why it's the preferred route because of the overhead involved in our case, on our part. So we're prepared to make it work, but then it really needs to be justifiable for us to consider it. And also keep in mind that the project lead will likely be liable for tax on the grant because in this case, it's going to be considered an income. So we do encourage that if you absolutely have to go through this route, you include the amount for taxation as part of your budget. We have tried to set up some mentorship as part of this process just to increase the quality of submissions received, but also just to benefit the wider Indaba community. So there is pre-submission mentorship, let me call it that is currently ongoing. There's a form which you can find on our Twitter page, and that allows you to ask for mentorship. If you'd like feedback on your budget, if you'd like to have someone read through your draft proposal, the Indaba committee is taking time to help the quality of submissions that are finally received. We also have an info email address if you'd like to ask any questions ahead of time ask for any clarification. Teams that are awarded grant funding will be assigned a mentor. So ideally, this will be a mentor who is an expert or more experienced in the field of your project implementation. and they will be available to you to meet up and catch up with as often as you need. So you can feel free to email them every so often. But then we will set up a midterm evaluation for each project, and this will be at least one instance where you will sit down with your mentor, have them assess how far you have gone with the project. So that assessment is going to be based on the deliverables that you set up as part of the application process. And then if any challenges have been encountered, then ideally your mentor should be able to help you assess if the direction of the project needs to change, or suggest some solutions or recommendations for any challenges encountered. And I'm going to stop there. Thank you very much, and I'm excited about all this.
Thanks a lot, Kathleen. And now we're going to be basically opening up the questions to the panel. So don't hesitate to, by the way, post questions on the live comments, and we'll try to relay those to our panelists. But first, Kathleen, could you tell us more about what are the areas where people typically struggle when they submit an application, and how can they improve their submission?
Okay, thanks for the question, Karim. I think two main areas I've seen individuals struggle. The first is structuring the budget. There's normally a bit of a back and forth, wondering what can or what is a viable expense and what is not. So let me perhaps mention a few things that have come up. Getting a research intern who you pay a salary every month, for example, that's an acceptable expense. If you need a good laptop to execute on the project, that's a viable expense. If you are working in translation and would like to hire translators to create a data set for you, that's a viable expense. And then being able to be very detailed about it. So we would like to see you break it down into monthly expenditure, even though we are giving out the money in a lump sum. So I'd say evidence of having thought through the budget is definitely a plus for your application. And the second thing is the ethical considerations, thinking through and demonstrating that you have thought through how your project will essentially affect society. So the societal impact of your project. It would be great to have proposals that show evidence that you are conscious of the fact that you're not just developing technology in a vacuum, but can acknowledge that this technology is actually going to be deployed into the real world and is going to affect people. So think about how it's going to affect people and how through your development of your solution, you can mitigate those effects. Karim, you're muted, so we can't hear you.
Yeah, can you hear me now? Yeah, I was saying thanks a lot, Kathleen, for the tips. I want to emphasize the fact that we have funding available. The deep learning Indaba team and all the partners that we have really delivered on that front. So please don't hesitate to apply, and you can also send us messages and request more tips and things. But I believe there is a great opportunity. It's not theoretical; it's very practical. There are some deadlines though, so make sure you apply very early on, and you have, as I'd say, hopefully a good likelihood of getting grant money, so that you can spend time on the challenge or on other research projects. It's not limited just to the challenge. So we have one more question. If I don't get selected for funding, can I still participate in the challenge, and can I apply for funding from other sources? Yes, absolutely. The participation into the grant challenge is open to everyone, and given the quality of the purpose and the goals and the partners, I think you are highly likely actually to get potential funding from other parties. So don't hesitate to take the initiative and contact us also, if you need advice. By the way, as was mentioned, we are opening a Slack channel to be able to follow you during this grant challenge, answer your questions. I'm sure you'll have many tactical questions. We have a roundup of basically people who are going to be mentoring, both experts such as Marcin or Ben, but also other mentors in machine learning and other fields. So don't hesitate to subscribe to, the workspace, like the Slack workspace that we will be preparing. And we can show the link here on screen as well to get people to sign on. We have also questions for Ben. So Ben, you got us all excited by mentioning that you had visual data. So could you tell us more about the data you have and how we can collaborate with DNDi to accelerate, converging towards a good outcome? It's also interesting because particularly on the Zindi platform, there is potential also to do visual AI competition. So maybe there's something there. So could you tell us more? Yeah.
Yes, sure. So one of the data sets of images that we have that I think is very interesting is from a process that we call high content imaging. So when we're looking experimentally to see whether a chemical can kill a parasite, what you do is you take cells, usually mammalian cells, and you infect them with the parasite, and you can take images of that. So what you'll see is the cells, which are like little circles, and there'll be little dots inside, which will be the parasite, and the number of dots will increase over time, and you watch that. And then you add your chemical to the effectively test tube that those cells are sitting in, and what should happen if the molecule does something good is the parasites should disappear, and the cells should remain untouched. And you can look at how that happens over time using imaging, and also you can look at how that happens over concentrations of the amount of chemical that you put in. And we take images of all of those different things. And usually we look for chemicals that are good at killing the parasite. But there's a lot more information in there. There is information like, do the parasites all disappear at the same time, or do they disappear slowly? Do they disappear some instantly and some over a long period of time? And if so, why? Is there something linked to the particular shape of the cell which can be correlated to the way that parasites might disappear? Also, one of the things we see is the cells can sometimes change shape during this process. They might not disappear, but they might change shape. And if they change shape, is that characteristic of the way that the parasites are disappearing? And all of that is incredibly rich in terms of understanding how the particular chemical is killing the parasite. And it may well be that there's a certain way of killing the parasite that is better and would lead to a more effective drug than others. Or it may well be that when you combine two different drugs, you get a different effect, and there might be a particular, what we call phenotype or readout that we're looking for. Those images we have, it's in the millions, all from the same machine, all taken by the same team at the Institute Pasteur in Korea, and where the data set is looking at different chemicals that come from different places, some that you can buy off the shelf, some that are the lists of molecules that Marcin mentioned earlier, and some of them are proprietary molecules coming from the vaults in pharma companies. And it would be really interesting to see if we can somehow link up the images, the image analysis coming from all that data over time, over concentration, and so it's multiple images in a package, to the nature of the chemical we're testing. Where did it come from? What's known about it? There's a lot of metadata we can attach to it. So that's Ben, one particular data set. Yeah.
Yeah, we have a question from the audience. So how do you annotate those images, for example?
Oh, that's beyond. I'm not sure I would be able to give an answer on that. If you, I can come back to you with an answer to that.
And importantly, would DNDi be interested in collaborating with the Indaba as part of the Grand Challenge to work on this? Because for us, I think this is exactly what our goal through this initiative is: bring people with significant domain expertise, such as yourself and your colleagues at DNDi, working with some of the very best machine learning engineers in the community. And I believe actually lots of positive outcomes can come out of this. So do you think this would be possible?
It would be possible. The question is whether we can. The question was whether we can prepare the data set in a reasonable time frame. We have the approval from most of the providers of those compounds, or those chemical compounds, to use at least the images, maybe not the chemical data behind it, or maybe a restricted amount of chemical data, because some of those are proprietary molecules. But in theory, that data set can be used. But we just need to compile it into a way that we can release into a public domain or a public forum like this. But yes, I can look into that.
And there's also exciting developments in machine learning around unsupervised learning. So you wouldn't necessarily need to label everything to get good results. So I think, like, this is definitely an exciting time. So we're happy to follow up the conversation with you. We have more questions coming, which is great. So for example, to Marcin, how do you define a PDB file?
Right. PDB files are quite well codified, so they have a good syntax, but they can also be generated by the scripts that you will have. So you can download them from our repository. They can be generated by the scripts. Like the structures can be predicted by online servers. And well, a PDB file kind of stems from the Fortran age, so it's column-based. It has coordinates of each and every atom in your system, and it has coordinates both for the protein and for the ligand, for the small molecule that's your prospective drug. And well, we will also provide a link to the formal definition of the format, just so that everyone is on the safe side.
And there's the follow-up question, which is about what are the good materials that you would recommend for a beginner who's getting started in bioinformatics?
Yes, so we'll also provide links to these, but for example, there is I can wholeheartedly recommend DeepChem. It's a library from Stanford that implements a lot of bioinformatics and cheminformatics tools for machine learning people. So it's I believe it's now either in TensorFlow 2.0 or in PyTorch, and it gently walks you through the whole process. So there seems no knowledge of machine learning or computational biology, and I believe in 17 notebooks you're good to go. You can do more than that, and we'll provide the links to these tutorials as well. But that's a good place to start.
Cool. Thanks a lot, Marcin. And we have a question about IP, intellectual property, for this challenge. So in this particular case, actually, this is something we're working on, but we'd like the IP to remain with the participants that came up with something. But also the goal of this is to actually accelerate, drug development and share with the community. So we'll try to find a way to make the results of the competition available such that partners can actually, go to like experimentation, like real experiments and potentially clinical trials. So just wanted to mention that we will be coming back to you regarding IP, but the goal is to be collaborative, and definitely it's not a predatory approach in any way. We had a question for Elaine also. Elaine, what have you seen in terms of like new technologies to address sort of like the natural tension between privacy of data and also progress in machine learning?
Yeah, that's a big question. I don't know if it's been so much about technology as it's been around discussions. People coming up with new ways in which we can think about data, and also be more aware of the issues with data, because it's very easy to get a data set, especially if you have a big data set, and just assume that it's going to solve all the problems that you want to solve without thinking about who is represented in that data and who's been left out of that data, and what are the issues that would emerge if you use the data as it is without accounting for those limitations. So I don't know about technology, but I think there's been a lot of discussions, and people have come up with a lot of ways in which we should be thinking about those problems and trying to address those problems. Karim, you're muted.
Yeah, could you tell us more about also what is happening, and that you're seeing, because this is a very current question on machine learning with coronavirus. So do you think there are great opportunities and initiatives out there, or what's your take on this?
Yeah, I think there are lots of great opportunities. The opportunities around disease modeling and forecasting. So a lot of the models that we tend to see in the news have been mathematical models, but I think that machine learning models also have a place to complement those mathematical models. And based on previous experiences modeling things like influenza, we find that mathematical models tend to do well when we're doing long-term predictions, and machine learning models will do better in short-term predictions. And so those are things that you can combine together. There's also been a lot of work being done around disease diagnosis as well. So how do we take images of the lungs and try to diagnose pneumonia and disease progression over time? There's been work also around telemedicine. So how can technology help improve telemedicine since we're all stuck at home and people are not really going to the hospital? I have colleagues who are doing work around cost effectiveness. So how do we make the most of distribution of drugs using machine learning and other tools? How do we make sure that, especially now that we have cases increasing in sub-Saharan African countries, how do we make sure that the hospitals, as the hospitals fill up, that we're not having a lot of deaths, that we're managing the cases properly? So those are some examples.
Yes, absolutely. We had one more question related to the Grand Challenge initiative. So, from your experience, have you seen a similar initiative in other regions, like targeting like finding a cure to a known disease that hasn't been solved yet? Like, is this something new? Like, what's your take on it?
I think this is new in terms of finding the cure, at least to me. I know the Gates Foundation runs a lot of grand challenges for different things, from looking at developing better tools for disease diagnosis, looking at vaccinations and other things. But I don't think I've seen one for finding a cure for neglected tropical disease. So I think this is pretty new and I'm very excited about it.
Cool, and indeed, like we haven't seen anything on our side as well. So I think this is super exciting moment. Our community is coming together. I mentioned a few people on the chat actually are saying hi from Tokyo. So the African machine learning community is really united around what we can do together, and so it's a super exciting moment. Celina, maybe also from your own experience looking at how Zindi, which is the largest data science platform in Africa, growing. What do you see are the opportunities, and do you see movement around these new novel ways to tackle problems?
Yeah, definitely. I mean, and health is really a big topic that has been coming up recently for obvious reasons. And I think that, as was touched on earlier, I mean, there's so many angles and ways that AI and data science and machine learning can play an important role. So actually from Zindi's side, we've had a number of challenges that tackle things like, from mental health to, broader public health to COVID as well. We collaborated with AI4D on a COVID modeling challenge as well. And now, because this topic is becoming more and more relevant and more urgent, let's say, I think that there's a lot of interesting data sets that people are interested in applying machine learning towards, including like contact tracing and things like that. So yeah, hopefully there's a lot more to do.
No, and it's exciting because everything is 100% scalable. So that means that we really can tap into the breadth of expertise in the community to tackle those problems. And you never know, you could have a positive surprise and an unexpected event happening. So by fostering this type of initiatives, I think definitely we're very modern in our approach. And as we mentioned earlier, like very different from other, classical initiatives. Curing disease is typically the realm of like either governments or big pharma. Like tackling it from a community and startup angle is something really genuinely new, and we think it's just the beginning. So it's super exciting. So we have more questions related to exactly like, how this competition would work. For example, a question for Ben from Jeremiah: how do you verify a molecule designed by an AI algorithm is a potential drug target? Ben.
It's a great question. So the first thing that we would do is we would get a sample of that molecule and we would test it to see if it can kill the parasite in a cell. So the images I was discussing earlier. So we would check that, and if it works, and particularly if it works well, i.e., you don't need very much of it to be able to kill off the parasite, we would then start looking at it in a variety of different ways. And I would term this as classical drug discovery. We would see, does it have any flags that would mean it could be toxic? Now, if your molecule has come from one of the libraries that Marcin was mentioning earlier of known molecules, then we don't have to test that because it's known. You would also check to make sure that it works in other models, maybe more advanced models, looking not just infected cells, but other cultures. And there's a whole process, a whole cascade that we have in place at DNDi and others have in place that basically chaperones the molecule through to first in human trials, and then you test it in humans to make sure it was safe and to make sure that it works as a pill and that it ends up in the blood and ends up where it needs to go, and then you would test it in patients. And then if it works in patients, you would take it for it to be registered. I've made it sound very easy. It's actually very complex and quite challenging, which is why I think I mentioned on my slides, we need about 20 shots on goal to be able to stand a good chance of getting one into the hands of patients.
Which is why this approach about having a scalable platform and tapping into the community could be a useful one, because you need indeed multiple shots to get something. So we have a question. We're almost wrapping up, but we'll still take two more questions. So a question from Daniela: I am a machine learning researcher and have little domain knowledge in drug discovery. Are there people in DNDi who are interested in working with me, and how to reach out to them?
Yes, there are. It's me and a couple of my colleagues, but mainly me. My Twitter handle, I think, is on the YouTube link, but I'm relatively easy to find online, and I would be more than happy to speak to anyone who's interested in contacting me directly.
Cool. Thanks a lot. So yes, I wanted to mention, like, really don't hesitate to reach out to our panelists and to the Grand Challenge team. Importantly, you can do this easily through Slack. So we will be sharing the links, several times, including in the Discord. Yeah, we can see it here on the Slack workspace. So maybe if we have one more question before we wrap up, so don't hesitate if somebody has a question. Yeah, we have one from Muad. Is there any way to evaluate the secondary effect of the new molecule, like toxicity and cost of manufacturing?
Yes. So so first and foremost, our molecules, at least the starting set, have already been evaluated. So they are non-toxic, they are drug-like, and they are inexpensive. So for most of the off-the-shelf molecules, the cost is relatively low. If you design a new molecule, then the cost can get arbitrarily high, because you have to sometimes invent new synthetic pathways, and these things can take a long time and a lot of labor. However, it's for toxicity, we have some rules of thumb that we can apply. There are machine learning models that predict things like toxicity. So yes, you can certainly do that, and we are doing it in evaluation, but sort of behind the scenes for the molecules that bind well. Because you can still work on a molecule, you can elaborate it so that it's make it less toxic. If it works very well on the parasite, you can still, there are things you can do to make it sort of less dangerous to the patient. So we first want to care for the efficacy, and then we can perhaps work with the toxicity and the transport, etc.
Thanks, Marcin. Also, important mention from Data Science Nigeria. To be very clear, there is a difference between the Indaba Grand Challenge itself, which is a very focused, project on curing Leishmaniasis, and the Indaba X, AI for Development Innovation Grants. So you can have a grant and apply for a grant for the Indaba Grant Challenge through, the Innovation Grant. But if also you have another research project which you're passionate about, you can also, apply for a grant in this area. So it is not limited to the Indaba Grant Challenge. So, just wanted to mention this clarification that Data Science Nigeria has put in live comments. And the other point we wanted to make is this is pretty much a starting point. We believe lots of great outcomes are possible through an initiative at this scale. Again, this has never been tried before, and certainly not at the scale at which we are talking, literally leveraging, talent from the continent in a scalable way with experts working together. So we're very excited about the challenge, but importantly, if you see ways where we can improve the process, please give us your feedback. This is pretty much a work in progress. I want to thank all the volunteers who have made this possible. Some of them are, the panelists today, but there are many key people behind the scenes who've been working tirelessly, sometimes pulling an all-nighter so that the competition is ready. So please make sure you register to the challenge, ask questions on the Slack channel. We will be there and available to help, and looking forward to interacting with all of you. This is a very exciting time, and I believe that we have a chance to make the world truly a better place and save lives. It really depends on what every one of us here collectively does to support the community and help people in need. So let's push super hard for this, and we will be available. And one of the ideas we had from this call is we'll also do regular check-ins with you. Particularly Marcin will explain in detail how the challenge works, where you can find the information about, basic bioinformatics to get you started. So don't hesitate to reach out. And thanks again to all our volunteers, our partners also, both African startups, Zindi, InstaDeep, and others, IDRC, SIDA, AI Knowledge for All, AI4D, who are participating into funding this, and also our compute partners such as TensorFlow, and hopefully we're going to onboard many more partners. This is a starting point. So thanks a lot for following and attending this live event. Hope you found it interesting and exciting, and we look forward to catching up very soon. Yeah. Thanks everyone, and talk to you soon. Bye-bye. Bye-bye everyone. Cool.